मैं किसी से बोलूँ चेक करने के लिए तो वॉल्यूम आ रहा है कि नहीं तो अगर हम से बहुत करें मैं वॉल्यूम फुल कर देता हूँ तो now may I request Professor Madhu ma'am to
Thank you so much. ये इंटरनल तो बंद करना चाहिए ना?
नहीं सर ये रूम में वो करना है.
I know. वो ले लेना.
I come for this NP view.
100%.
I have to go somewhere.
I know you speak. We just spoke.
Thank you.
So I can now start, right?
Just 5 minutes.
Right.
I will request Alia from Shivnata University. So this time we have a ritual that students are introduced.
Very nice.
So that they will also have the practice. So Alia from Shivnata University, she will be introduced.
Good.
So good morning to all the professors and my dear friends. It is a great pleasure to introduce our speaker of the day for today's seminar, Professor Natarajan sir. He will be talking about a condition which is caused by Mycobacterium tuberculosis and how it affects our lungs. This subject is of great interest to me because years back I was also infected with the same bacterium. So I'm really looking forward to learn a lot from the seminar. It is my great pleasure to introduce Sir, who had obtained his bachelor's in botany from University of Delhi. He also did his master's from University in Delhi as well in botany. He has worked at various organizations. He had been a part of. He had been a research scientist at National Institute of Immunology in New Delhi. He did his postdoctoral fellowship from University of Texas MD Anderson Cancer Center, which is situated in Houston. Shortly after that, Sir worked as a PI in Lupin Lab, which is situated in India. Proceeding to that, he has also been a part of International Center for Genetic Engineering and Biotech as a PI and has been part of ACBR since 2010 and is still there. His current research interests are immunology of infectious diseases, including Mycobacterium tuberculosis, human immunodeficiency virus, Plasmodium falciparum, and Streptococcus pneumonia. During his bachelor studies, he worked on a thesis which was about response of Rhizobium strain DDSS69 to sodium chloride stress. His achievements have been recognized by many different awards and honors. He has been nominated member of the DBT Task Force on Infectious Diseases Biology in 2010 and is part till date. He was also a chairperson screening committee of Twining R&D program for Northeast region in 2016.
Enough. We read into my lecture time, no? That's okay.
So just 2 minutes.
No. I'm here. I lost my way from botany, ended up in TB.
Okay, so everyone please join me in welcoming Sir.
Thank you, Aparna, and thank you, Meenakshi. No, I remember your name, Meenakshi. Meenakshi was the first person I met when I joined ACBR. The two of us were very obedient people sitting in one sofa waiting for the director. She was joining as a research associate. I was joining as a professor. Okay, so she is now a wonderful colleague, is part of that, and then along with Aparna, of course. So, as the student very kindly introduced me with some very nice words. She said that she was a part of TB, and when you said that, I was really looking forward to. I held my breath. I don't know what you were, another infection, or to my talk. I was a bit confused that way. Okay, so let it be a two-way street, right? So this is enough. I don't think I need to go full screen or something. Right? This is fine, and I hope you are able to hear me through the N95. Right? Still not out of the woods, and we'll talk about it a little bit, right? So here's the title: How do you solve a problem like Mycobacterium, the Immortals of the lung? So you see, I've underlined two words here: problem and the Immortals, right? The problem is basically inspired by Sound of Music. If you have seen the classic, there is this very nice song called "How do you solve a problem like Maria?" Maria is a nun who actually joins the covenant, but then later on moves. You know the story? Sound of Music classic नहीं पता पहले तो वो बोलो उसके बाद ना it's a classic it's about how a family which was into music migrated during the Second World War, right? And then there was a nice movie. So and she is a troublemaker in the nunnery, and then they all sing a song like how do you solve a problem like Maria. So I borrowed that title. The Immortals comes from the way TB infects us, and the Immortals of the lung is also inspired by the trilogy by Anish Tripathi, Immortals of Meluha and the Shiva story, right? If you have read the books, it's a nice trilogy, 3 books he has written. So why do we say immortals, and why is it a problem? The problem is, like we all had infections since our birth, right? We've been vaccinated for quite some time. You people have been vaccinated more than me. They have been vaccinated more than you. Some infections have been eradicated. Like I've been vaccinated by smallpox also when I was a kid. Luckily, it's been eradicated because of very nice vaccination programs, right? And every day, like there's something or the other. I got the chicken pox, but you guys, I'm sure you guys have been vaccinated with chickenpox vaccine also, right? So now the problem is we have been bombarded with infections right from our life, and like the tropics, especially India, is home for all the infections. Well, all the infections are here, right? And the West normally deal with things like cancer, lifestyle diseases, diabetes, and all. Okay, it is also a part of India now. Like India is a diabetes capital simply because we started aping the West, less exercise, more junk food, etc., etc. Juvenile diabetes is a major problem in India. Okay, along with cancer, of course. Like we have had probably nice talks on cancer, or will be due. Right, Dr. Raman, who spoke last before me, is also works on cancer. So you had a very nice Golgappa. If you haven't, please visit the Patel Chest fellow. There is a guy who's older than me and been selling this chaat papdi and all, right? And a few flies will also be there. But then if you ask somebody, I say it adds to the taste. Doesn't matter, right? So you come down with E. coli infection, you come down with salmonella infection. It comes, it goes away. This chap is different. Mycobacteria is different. Once you get infected, you remain infected for life. There's hardly anyone in this room who's not infected with tuberculosis, right? So, but then it doesn't kill us. COVID, if it infects, if you're not treated, if you don't have a strong immunity, that's the catchword. You'll probably end up in a hospital, and unfortunately, many millions of people have lost their lives. Now, TB doesn't kill us, just like many other bacteria which remain in our upper respiratory region. Streptococcus, Staphylococcus, Corynebacterium diphtheria, all the unfriendly guys simply have our home here. Why is that? These chaps have evolved to infect humans. There is no natural animal reservoir for any of these infections. We do mice experiments, like that's why it's known as experimentally induced tuberculosis in mice or rabbit or guinea pigs. They're not natural hosts. If you give them, they will take it. And mice is resistant to it. Like they can take bucket loads of this bacteria, whereas guinea pigs are a little more sensitive. It infects, but it doesn't kill. That means what? It's like a ticking time bomb. So it's a problem. Like we don't get what is known as sterile immunity. So once it goes in, like after Plasmodium, malaria, salmonella, you take drugs, it goes away. Till you are bitten by another Anopheles or you have another Golgappa or something. But TB, it's in the air. You breathe it in and you breathe it out. And how many bacilli do you actually require to establish a primary infection in lung? 5. 5 bacilli kya hota hai? If you take an inoculation needle, like you've done some experiments in microbiology out here, you'll have millions of bacteria. Imagine 5 bacilli, you can't even see. 5 bacilli, I'll have a cartoon somewhere on the slides. 5 bacteria are all that are required to establish a primary infection complex. It has been experimentally proven. Okay, so that means what? 5 bacilli, breathing 5 bacilli, and lung is a very sticky. They have surfactant proteins and all these steps are there. So it will stick and establish a primary. Where it goes, we will talk about it. It goes to alveolar macrophages and makes it a home. So that is the mother of the problem. Okay, so as a result of this problem, what do you have? You have this. This is the TB 2020 statistics of tuberculosis from WHO. 2020, the TB cases went even higher simply because most hospitals started reserving beds for COVID patients. Right? TB to chalta rahega, inko bachao. Right? Which is sometimes like you have to make a judgment call. So a lot of these diseases actually got neglected also. The TB, and look at India, it's not a very nice color to see. And this is the mortality in 2020 in TB. TB kills 1% every 8 minutes. By the time 1 hour passes, I finish my talk, many people would have died. It's a major problem. 8 million new cases per year. That is the general statistics. And Once it infects, it doesn't go. And there is also, like something like a belief. Nobody knows. Really, unless you totally dislike that person, you cannot cut open that person and then see whether TB is there or not. Right? So that is the case. So you think that like if it's there, high bacilli, sticky bacteria, it goes there, stays inside, it's there. So this is the problem. So what do we do? In addition to the health costs, these are some of the indirect costs and direct costs to the society. Look at that. Productive work lost because of TB illness. Earlier it was thought that only the poor people or people from the lower economic strata get infected with TB. No longer the case. Even celebrities have been like victim for this. Anybody can get it. It's in the air. Anybody can get it, right? But then it was like, but then if some a well-to-do person gets infected, he can take a leave from work and all. A poor laborer, daily wage laborer cannot do that. So it's way lost. School dropouts due to parental TB, very unfortunate. Right? Parent gets TB, the kid is thrown out of the school. And the last is even more disturbing. Women rejected by families due to TB, basically by in-laws. Okay. So these are what are known as indirect costs to the society. So it just is not reflected only into that person. It has a wider ramification. Right? So these are some of the things that one needs to build in when making policies. Like TB is a national mission. We have ICMR, that's the Indian Council of Medical Research. These are regulatory and sometimes funding bodies. And Department of Biotechnology, somebody like she said that I'm a member of the TB task force, also I'm a member of the COVID task force. So tuberculosis task force and infectious task force, their mandate is to generate policies, to find like avenues for grant proposals, like which direction to go. I'll talk about it in a minute or so. So this is the problem. Okay. So this is what like you have started, and it's there at the global level. Like there was a global burden is like this much. What they want is 2050. There should be at least one case per million. And this is the latest by Department of Biotechnology, which comes under the Ministry of Science and Technology. This is initiative was nice launch of Dare to Erad. They have these acronyms. A data-driven research to eradicate TB. What they want to do is they want to get people to come together and do research, like build teams. Rather than me in my own small lab working and doing something, and then Aparna in 2 floors down working something, and Meenakshi something, why don't we join hands? Aparna may come with an expertise, I may have a little bit of expertise, Meenakshi may come with an expertise. Meenakshi may be a genomics person, right? I may be an immunologist. She may be she works a lot on lipidomics, so lipidomics person. Why don't we join hands and make a small project? These are all known as program projects, or they call it centers for excellence. Multi-pronged approach, right? To treat tuberculosis. It's not just treat, there are a lot of problems with tuberculosis. This is the case. We have it in our lungs, right? And I said it cannot take out. So it's like a battle of chess. So the question is who will blink first and who will say checkmate. And luckily we are since alive because we have Ishi Anand on our side, right? The lung actually, and then since the white ones, the person, if you play chess, the white pawns are the ones that will like get a precedence over the black one. They make the first move. As long as the immune system is healthy, like us right now, the scales are balanced. The TB doesn't kill us, but it does not allow the immune system to throw it out of the system. It's like a forced symbiosis. We have no choice but to live with it. Just like people are now saying that we may have to live with COVID. It's not going to go away soon. It's a virus, it's an RNA strand, positive strand virus. It will mutate like crazy, just like HIV does. So it's not going to go away so soon. We may have to take booster shots. Hopefully you're all eligible, and then please go and take the booster shots. Okay. So, but then the moment immune system takes a dip, this chap surfaces. The scales get tipped in front in favor of tuberculosis. Can you tell me when this might happen?
In case of immunodeficiencies.
So immunodeficiency किस way होगा? Can you think of a pathogen?
Getting an HIV infection.
HIV infection is one. So if you go back to this, most of the TB in the African subcontinent is because of co-infections with HIV-TB. We also did a little bit of work on this. Okay, but TB mostly in the Indian subcontinent is HIV free. It's pure TB. And that means what? Depending on where you are and what kind of tuberculosis patients infections, your management and treatment strategies will be tailor-made. What works here may not work here. Look at this. These are all quite clean, much better than this thing. Why is that? Despite the fact that BCG vaccination is mandatory in India, right? That's probably you may have seen a little scar on your upper left shoulder, right? The arm, right? There's no TB, there's no BCG vaccination out here. There's no mandatory. It's there. It's not there, but it's still there because they say there are several reasons. Geographical distribution is there, but despite that, if there is a vaccination and it is given on day one of your birth, the day you are born, by that day or the next day, the pediatrician will come and give you the vaccine. You're like, it is a vaccine which is given only by the pediatrician because it is live BCG in an emulsion and it is given right under the skin. It's a subcutaneous, no? So it requires some training. Nurse is not authorized to give it. So the vaccination is given by your pediatrician only. So 50 microliters is inserted right under the skin. You can actually see that 50 microliters. The baby's skin is very thin. Okay, you can actually see it. I saw that being given to my daughter when she was born. So BCG actually works as a very nice immune potentiator or immune activator. It stimulates the immune system. You know, neonatal immunity, when you're born, neonatal immunity is just about beginning. And just like that, in the old people also, there's also known as immune senescence. That means aging of the immune system. Somewhere between, let us say, 15 to 20, and to about 55 to 60, the immune system works very nicely. Later on, there is immune senescence for several reasons. We don't get into that. And this is, of course, developed. BCG gives excellent protection till puberty. Till the time, like the child is about 13, 14 years, BCG gives very nice protection, almost 100%. But when puberty kicks in, the person is as susceptible to TB as an unvaccinated individual. Okay, several reasons. Why? Because it's in the air, so BCG gets thrown out, and then you have TB colonizing it, and then establishing fresh infections. All this is there, but that is not the case because environmental mycobacterium load is very low in the western ocean. Of course, with migration, it became a problem. West was never interested in TB. They thought that this is a poor man's disease. Let them deal with it. We'll deal with cancer instead. HIV actually came to the rescue of tuberculosis because once HIV started, and HIV was it got more prevalent in the West. People with weak immune system, like Shivam said, TB started resurfacing. That's when they realized that, oh, we already carry it in our lungs. It's just that we didn't fall prey to this disease. We don't get symptomatic disease. That means that we didn't know that it's already there. Then they realized that okay, TB is a major problem, and that's when NIH, which is equivalent to your ICMR, that's National Institutes of Health, which is a major funding body. So they started funding more programs. So then the collaborations and all. That was like almost 30, 40 years to make it straight up in that. So this is the problem. Okay. There's three important species which are of use to us: Mycobacterium tuberculosis, which causes Mycobacterium pulmonary tuberculosis, and extrapulmonary. There is hardly any organ except the heart which is not infected by TB. So look at it; it is so smart. The moment it infects the heart, the person will die. And since it has evolved to infect humans, it doesn't want to kill the host. So except heart, including from the brain to the toe, everything, all organs in between will get infected. You have kidney TB, liver TB, genital TB, intestinal TB, bone TB, even the meninges get infected with tuberculosis. Not the brain part, but the meninges get infected with tuberculosis. Lung, of course, is there. Yes.
So why not heart, sir? Like, sir, heart must be also containing the receptors. And sir, how
So,
that shouldn't infect this?
So most cardiac muscles are divided of most receptors. They are system player. And people and almost even HIV doesn't go to the heart. Well, HIV doesn't go to the cardiac muscle anyway. It remains in your monocytes, macrophages, and basically your T cells. None of the bacteria are go inside the heart. If you take a cardiac muscles cannot be cultured. Right? You cannot culture cardiac muscles. So even if you, let us say, take cardiac muscles like you make a monolayer and add in nothing because they are totally unreceptive. That's one. So probably our own system in the evolution has made sure that the heart is a what privileged organ? It's immune privileged. Yes, immune privileged means that the immune cells are generally not required there because inflammation was there. Yes, right? Just like the eye is also known as an immune privileged organ. Right? It's a misnomer because privilege means you would want something, but they have the definition the other way around. Okay. But of course, we have the other one called Mycobacterium avium intracellular, or also known as MAC complex. This is there mostly for like in HIV associated TB. Then leprae, which causes leprosy, hydromatus and lepromatus leprosy. There's a lot of homology between these three aspects. Like they have like 98% sequence homology between them. Okay, so let's deal with it. So these are all the other strains of Mycobacterium which are present in the environment. Right? Sometimes, like for example, if you see, you have Mycobacterium. I had introduced one more slide. I don't know where it went. Mycobacterium marinum is there. Okay, it causes fish TB. In fact, it's there what is known as aquarium TB. People who have aquarium, if their fish gets infected, they can get aquarium TB from the aquarium. Okay. Then there is M. bovis, which basically is the parent strain for your BCG, which is M. bovis BCG, attenuated strain of M. bovis is M. bovis BCG. So calmette-guerin, BCG stands for Bacillus calmette-guerin. These are the two scientists who passaged M. bovis till it lost its virulence, and then formed the vaccinated strain. This was almost 200 years ago, and till now, that is the only vaccination available. People have tried. I'll talk about vaccines. What are the different ways by which you can vaccinate, improve BCG, and the other strategies for prevention and treatment. Okay. AVM, of course, is HIV avium. The reason why this is a problem is because it has a hugely complex, multi-layered cell wall. So this creates a major problem for the drugs to penetrate. Now, when you talk about latent tuberculosis, that means what we have, right? Infected but not symptomatic TB. So what that means is that what is the bacteria's pathology and what is it doing there? It simply goes into some kind of a hibernation. So in the lungs, what they have shown is that this TB simply stops all metabolic activity. It stops breathing. It goes to an anaerobic, switches to an anaerobic respiration. It doesn't use any oxygen. There is no protein synthesis, no metabolism, nothing. This is where the problem comes. All the drugs that are there for tuberculosis, they are against actively metabolizing bacteria. How do you do a drug discovery? You grow, you find a target, and then you find a molecule to bind to the target. If this thing just doesn't grow, it's not doing anything, what do? You do to a cell wall, it will laugh at it. He says that I have so many layers of cell wall, be my guest, nothing will. So that means what? Even these drugs that you use to treat TB, they have to penetrate all these layers and go to a target. And what are the targets that we have? We have rifampicin, which is an RNA polymerase. Target is RNA polymerase, right? Pyrazinamide, which is again a mycolic acid, which is part of your cell wall. So जब cell wall synthesis होगा, तभी तो वो mycolic acids बनेंगे ना. The cell wall synthesis ही नहीं होगा, जितना मर्जी डाल दो. Add a kilogram of pyrazinamide, nothing will happen. Similarly, ethambutol. Then you have the gyrase inhibitors and all, but all those will come when you have DNA replication. So nothing is happening. So treating latent tuberculosis is a major problem. People have billions of dollars have been put into this, but with no success. You see sporadic publications here and there that they have found, but you take it from mice to guinea pig or to any other human trial, it falls flat. Same is the case. Remember, this is a case of COVID. Just take the case of COVID. Bacterium is much more complex. It has thousands of genes. So viruses have much lesser genes, a handful of genes, 10 to 12 genes are there. Okay. HIV, same problem. But HIV, thankfully, over the years, we have perfected the what is known as heart therapy. Okay. So use the code. It will take a while to load. On double click. Okay. This is highly active antiretroviral therapy, or HAART, heart therapy. Okay. Because of that, HIV cases are really come down. But let's take the case of COVID. There is hardly any lab in this entire world which have not started working on finding a drug for COVID. It's been 2 years, nothing. We have tried many things, but nothing. We tried drugs from others, but on drug repurposing that we will talk about as we go along, even for TB and other cases. उसमें नहीं आया ना. So it's not easy, despite the fact that this virus just has 4 to 5, let's say 10 to 12 genes. This has 4,000 genes. License key not valid. Okay, one thing it's not valid. Okay, so this is the case. So now, if it, I don't know whether that slide is there or not. Okay, let's look at this. So there are 3 key aspects of dealing with any infection. Like, no, there's not TB. We need very good diagnosis. We need an effective treatment, fast treatment, and prevention, which is always better than cure. With TB, there is a problem in all the 3. That is why I said how to solve the problem like tuberculosis. The problem is not because of the number of cases. Of course, it's related. Why is the number of cases? Simply because of these 3. Let's look at diagnosis. Now, once you diagnose something, any illness, battle is half won. You know exactly which treatment to give. But in this case, not only is the diagnosis a problem, treatment is even problem. And BCG vaccination, we said that it gives you protection only in the childhood. In UK, the BCG gives very good, probably with the genotype. The 80% success rate. Here, the success rate is 0%. To remove the childhood part, right? Adult protection is 0%. Everybody falls flat on tuberculosis. You know, when you have TB, there is a lot of sputum inflammation. So what do? You take the sputum and then you look for the presence of this bacteria by what is ZN staining or acid fast staining. Sensitivity is 50%. You know, like Dr. Daman's talk, you have the PCR should have talked about these 2 terms, sensitivity and specificity. You know the difference between the 2? What is that? What do you, when I say sensitivity, what do you mean? What do you understand? Yeah, so when you say 50%, that means what?
Like half.
So if there are 100 people whose samples you are testing, only 50% case may that sputum is there. All 100 may be TB positive, but the TB sputum, you will see bacteria, will see only in 50% of the people. That is known as sensitivity. That means you need to make it to at least 100% or at least more than 90% to have sputum acid fast staining for to show as a potential diagnostic tool. Second is the specificity. Specificity means that it should pick up only the tuberculosis. You know, when you have sputum, it may have other bacteria also. So if your PCR primers or let us say your staining techniques are not like that, like acid fast, there are many other acid fast bacteria also there. So that will also there. So specificity becomes a problem. Then, unlike E. coli, you know, E. coli, if you have isolated plasmids in BSc or like if you are doing your sub-training programs, you take a little bit of one loop and then put in a 50 ml flask, you put it in the incubator in the evening. Next day morning, it's full. It's completely turbid, right? It's grown like crazy. It takes 3 to 4 weeks. It's a very lazy bacteria. The doubling time of E. coli is 20 minutes. E. coli we normally use in the lab. This is 18 to 24 hours. Okay. So that means what? Once you plate it on a grow plate or you let's say put it in a culture, they will actually take 3 to 4 days. That is why whenever like my students, they do any of these plating experiments, they'll plate and then they'll go to their favorite god and start praying that nothing should happen for 3 weeks. Because this humid, especially in October, August, July, August, they go nightmare. Fungal contamination, spores and everything. Result आते उसमें contamination आ जाएगा, then all you need to do is just put bleach and then throw it. It's as simple as that. You cannot do it. That means what? 1 month experiment gone. और अगर वो mice का experiment होगा, everybody is comfortable with Hindi, nobody from south or anything. You're comfortable with Hindi? Switch to English. Okay, no, but we have foreign students also. So sometimes we do bilingual to make our point, but doesn't matter. So, what was it? Yeah. So let's say a mice experiment. You do your experiment, then after 3 weeks, you want to see what is the bacterial load in spleen, lungs, kidney, everything else. So you sacrifice my single cell suspensions. and then take and dilute it on this LB plates, not LB, the 7s11 plates, and then room for coliforming units or CFU. So that 1 month and this 1 month, 2 months gone. That is why PhD takes 5 or 6 years. Okay, so be careful. The other way is that you can do PCR. Right, the government is also developing a PCR-based technique. So this is the one that is being used, but it does not pick up all the strains. This is a transposon element, insertion element 6110. It doesn't pick up all the strains across geography, so you have a problem. Plus, 98% homology with BCG vaccinations. So there's a lot of cross-reactivity. So, and chest X-ray also, I didn't put it here. Chest X-ray also 50%. So how do you say something is tuberculosis, right? So you immediately do sputum, you put a culture, and then you look at X-ray, and then the other symptoms, the fever, the breathlessness, because it's affecting the lungs. So what is known as an empirical diagnosis is made. Empirical. That means the clinician has to take a call. Because the symptoms generally go with Streptococcus pneumonia, many viral fevers. By the time a person gets confirmed with tuberculosis, quite a lot of damage is already done. It takes almost 1 month. And then the antibiotics are put. Okay, we come to treatment. The antibiotic, let me finish these things. So how to solve it? Then they came up with what is known as MIGIT system, MGIT, or Mycobacteria Growth Indicator Tube. What does it? It can do fast diagnosis, right? It's based on a fluorescence marker. So what do? You add a fluorescence molecule, let us say a fluorescence indicator in the growth medium. And since the growth medium also has dissolved oxygen, the oxygen actually binds to the fluorescence molecule and quenches the fluorescence. When mycobacteria grows, it breathes. It takes in oxygen, thereby releasing the fluorescence. So this fluorescence is now taken up by the detector. So instead of 3 weeks, you can get this information in 48 hours. The backtech system is expensive. Not all.
Specificity will be less. Any bacteria growing with fluorescence.
Yeah. So you do sputum clearance, and later on, then once whatever grows, you do a diagnosis, like you do a PCR for that. So one particular MTB gene, you pick it up, right? Or a stick assay, you do it, right? Just like you have this stick assay, like a pregnancy test. That way you do it, so it is there. So it can do that. That's a very good question. So in fact, this kit was developed by Dr. Vijay Chaudhary from South Campus Biochemistry Department. So along with Backtech, now they have introduced that his kit is also there. Okay. So this, but then at least solves a major problem of waiting for 3 to 4 weeks. So the clinician within 48 hours knows that, okay, instead of giving a simple antibiotic like azithromycin or like ciprofloxacin, he knows he has to switch to a TB specific regimen. So, but then, you need to evolve science also. So a lot of okay, so this is what I told you, that the thing quenches and then the fluorescence is released, picked up by a fluorescence detector. Then the Gates Foundation, the Bill and Melinda Gates have this foundation where they give grants to innovative ideas. They say that give us out of the box. So this is also a new initiative called FIND, Foundation for Innovative New Diagnosis. So if you have a crazy idea, you can simply log into their website and say that I want to do this. And if you have a recognized, let's say if you have a permanent position and all, if they like it, they will ask you to send a small LOI, what is known as letter of intent, a short project page. Okay, like what you exactly want to do, how you want to do it, where you want to do it, and then they'll give you, let's say, $100,000 to $200,000 to start. And if you're able to do something good, they will actually give you $1 million to work on it. So, but it's highly competitive. It has to be really out of the box. It cannot be that so and so showed it in horses, I will show it in donkeys. Us type ka nahi hoga. So it has to be totally out of the box, and there's a very strict monitoring. Every 2 months, 3 months, they will ask you, like, what is the progress, what have you done? If you have, let's say, 3 objectives, so if you say that in first 3 months, I will finish one objective, they'll say if you have finished it, then otherwise they'll stop the grant. So it's highly monitored, very competitive to get, and you have to be really good at running the project also. Because they're funding a lot of money, so they want this thing. So this is a grand challenge. So based on that, this thing was diagnosed. So when you breathe, just before the New Year Eve, you have these alcohol detectors. The police uses near India Gate, cannot place, and all these are some places in Delhi. Okay, you can visit them. Good shopping. So you have this. So all these people will want to have some fun. They will drink and all and drive, which causes major problems. So the police have these alcohol detectors. Just like that, you have this known as an electronic nose. So when you breathe out, you release a lot of volatiles, right? So these volatiles, and they are handheld battery operated. So you have these cartridges into which this person will breathe. That cartridge can be sent to a GCMS, gas chromatograph, LCMS, and then you get what is known as a signature. And the board is not working for some reason, otherwise I would have, like, you see this. So in volatiles, if you do a mass spec, you will be this kind of a pattern. Okay. So for that, what you do is that you take these TB patients and then you take their confirmed TB patients when you develop this thing. You'll get one signature and then you do a test sample. Then you have to rule out some of these because somebody may be TB positive as well as streptococcus positive. So his signature would be different. So based on that, a lot of people have come up. In fact, there's one group led by Dr. Ranjan Nanda at ICGB. I spent about 12 years as a faculty there at the International Center for Genetic Engineering in Biotech. So there, he developed this using LCMS and gas chromatograph. He developed this thing. He was funded by the Bill and Melinda Gates Foundation. So there are ways to, and these are very fast diagnoses. Only problem is you need a gas chromatograph and LCMS. That means it is suitable in metros. But by and large, metros में ज़्यादा generally problem भी ज़्यादा होती है because there is a lot of crowding is there. No. So otherwise, if somebody is having a problem, he can always refer to AIMS as a tertiary care hospital, and then some things can be done. And meanwhile, and midget is something that can be done everywhere. So now, because this TB mission is there, so the government is funding. Like even in small tertiary, it may not be in every clinic in every village to begin with, but in one district you will have 2 midget centers, like back-tech centers, where if somebody is working, see there are what is known as TB hotspots also. No, not every area will have as many cases as every other area. So you have TB hotspots. वहाँ पे आप you have there you put a back-tech center, so fast diagnosis can be done, and the treatment is free. Problem comes in compliance. The compliance is, let me see if it is there. Okay. Before that, the diagnosis is चल रहा है. Already 11:15 हो गया. Okay, I'll finish the diagnosis and then we'll talk about how to solve the problem. Okay. 5-10 minutes extra I can take. Okay. So then this gene expert, it's a company, they came up with a PCR based for resistance. Okay, now before we go to resistance, let me talk about something else. Why we show that there is a problem in diagnosis. So what is the problem in treatment? Problem in treatment is even worse. Even if you diagnose somebody with tuberculosis, if you eat, again going back to the poor old Golgappa, you come down with an amoeba infection, you take 5 to 6 days of metronidazole or some tablet, you are fine. Okay. Tuberculosis, the treatment for a drug-sensitive strain is 6 months. The problem doesn't stop there. Problem is compliance. For the first 2 months, it is known as the intensive care treatment. The next 4 months are known as the follow-up. The combination of antibiotics is different here. The combination of antibiotics is different. The problem is, within the first 2 months, 95% of the bacteria are killed. Person feels that he is recovering. He stops taking the drugs for 4 months. This is the problem. That is why even when you come down with simple infections, if the recommended course of antibiotics is 7 days or 5 days, take it for the entire, even if you start feeling nice for it, because it is that 3 to 4% bacteria which are there, that is the major problem. Then they become resistant to these first line drugs. Okay, so you have single drug resistant, you have multi-drug resistance. If you are resistant to more than, if something is resistant to more than 2 of the 3 frontline drugs, known as NDR. उसमें problem solve नहीं हुआ, तो then you have what is known as XDR or extensively drug resistant. Even वो भी हुआ, तो now you have TDR, totally drug resistant. Nothing but totally fatal. Even XDRs are fatal. So you have TDR is a major problem. See how one problem leads to the other. One person gets drug sensitive tuberculosis infection, takes the drug for 2 months, starts feeling better, forgets the 4 months. Okay. Now, when this person is now harboring what kind of bacteria? MDR bacteria. Comes in contact with his own family member, right? Or traveling in the metro or bus or any place, coughs, aerosol generation. So this person gets directly infected with an MDR stain. The treatment for MDR is 18 to 24 months. From 6 months directly to 18 to 24 months. Major problem. Some of the drugs are psychotropic drugs. And most important is you have huge hepatotoxicity. These drugs are very highly potent fifth generation, sixth generation antibiotics. They have huge side effects. The liver goes for a six. Very difficult. That is why like the first thing the TB does is it kills your appetite. You don't feel like eating also. So, but then you need to eat to maintain your body. Okay. So then once the like first 2 months of intensive care case goes away, 80-90% bacteria are killed, person recovers. Like his fever is gone, spit is gone. You are nodding. I can understand. You've gone through this thing, so like you start getting weight gain. You start getting this that appetite. It's not easy to eat 4 to 5 drugs every day, right? So then this to stop this problem of MDR, the government initiated another kernel called DOTS, DOTS, directly observed treatment short course. That means what? It's not just the prescription is given. The prescription along with the drugs is given, and this healthcare worker, he or she goes to the patient and makes sure that they take the medicine in their presence. The medicine is not given to them. They will come and give you the medicine. उससे क्या हुआ? MDR cases really plummeted. Indirect benefit, it started generating employment. You see, the government has to worry when you make a policy. You need to worry about all these things. So employment भी generate हो गया, आपका cases भी कम. So this is the major problem in that. The problem is in treatment. What was prescribed in the 1940s, even before we became independent, it is the same drugs that are being prescribed even in 2022, barring only one, darolutamide, which is given to your HDR or MDR cases. Okay, that means what? 70 years or 80 years, we have not been able to find out a new drug for TB. Very unfortunate. Look at the breast minds in the world. They are like working on tuberculosis. They have all the technologies, but nahi ho pa raha hai. This bug is so smart. Just like COVID, 2 साल ही हुए हैं उसको, we don't have a drug. But then here we have 70 years no drug. But darolutamide hai. Problem is the misuse. For any small thing, the doctor will prescribe a huge 4th or 5th generation antibiotic, which can be treated with a simple ampicillin or something. The idea is to that इसकी डॉक्टर की एक दवाई में मैं ठीक हो गया, ये बहुत बढ़िया बात है. It's like using a cannon to kill an ant. No need, just stomp on it, it will die. But if you do cannon, if you keep bursting cannons, 1 or 2 ants will become resistant to cannon. Then what do? Then you have to bomb a nuclear bomb, then we'll all die. Okay. So this is one of the problems in the treatment. Okay. So we have covered diagnosis ka problem, we have covered treatment ka problem, we have covered the. So that is why this is a major problem. And because there is no sterile immunity, these are known as immortals of the lung. Okay. So Gene Expert works by a PCR sample, right? But the only thing is the cost is 1,500 per sample. Poor man cannot afford it. The laborer's minimum wage is 400 to 500 rupees per day. I don't know, 400 to 500 rupees per day. Whole day he will work, he'll place stones and everything. He will like lift his or her on the head a lot of bricks. They'll get 400. How can they have a 1,500 samples? So the Indian government, the ICMR and DBT initiated and found out TrueNat TB. This is Atmanirbhar thing, which is only given free in all government hospitals. Based on similar technology, picks up TB and also rifampicin resistant TB. Rifampicin resistance is the most prevalent drug resistance in the Indian subcontinent. So this is given free, and for private people it is maybe 250 rupees or so. So it is still manageable. So just like free vaccinations is there. So this is the diagnosis which is being actually being made to at least diagnose it. So how do you solve the problem of research? So before 1996, nobody had a clue. There is another thing with tuberculosis. If you grow E. coli, it grows within 12 hours, it will grow. It will become turbid. You centrifuge it to bring down the pellet, and then you supinate and you discard. But if you take supernatant, if you take and run it on a two-dimensional electrophoresis, everybody knows what's a two-dimensional page. The first dimension is based on pH gradient or charge, the PI, and then that strip you layer it onto the SDS page. So now the separation is based on the mass. So instead of bands on a gel, you will get spots. They are analyzed by a software, okay, because it's not easy to analyze like with your own naked eye. You hardly see few proteins. If you take a similar culture supernatant of tuberculosis, irrespective of whether it's early lab, log, and late log, there are differences. 3,000 to 4,000 proteins you can pick up. That means what? Tuberculosis secretes out all these proteins into the medium. Okay. Extrapolated into the lung. From the infected macrophage, it does exactly that. Right? So these are some of the evasion strategies that I'm now talking about. So when you have these strategies, the immune system feels that you have fantastic antigens, and that's what people also started doing. That okay, maybe some of these antigens we can use for diagnosis or for making vaccines. Kuch nahi aaya. Neither they made good vaccine diagnosis nor for vaccines. Okay, so some of our work also threw some light on this. That when we take these proteins, and they have very nice secretion systems. Each bacteria have very nice secretion systems there. Like it's much more complex in microbacteria. If you take any of these proteins, nobody knew. In fact, when you actually had a diagnosis, you have this Montu test. This is nothing but this culture filtrate protein. You do a 75% ammonium sulfate cut, resolve it, and then use it. Okay. So it was very interesting. When I was a student like you, and then I had to go after PhD, I had to go for my postdoc. So that time, the West were not at all interested in tuberculosis. So they used to take this as a sign of infection. Because we are vaccinated and there is a homology, if you become Montu positive, the doctor will say, you are protected. But if the same thing is read by an American doctor, he says, oh my God, you're infected. The same test, two different analyses. Then they really found out an HIV came to the rescue. Then they found out that, BCG, when this sequence came out, they found out that there is 98% sequence homology. So there are regions in this tuberculosis which are absent in TB. They are known as regions of difference in the BCG. They started picking up those regions and to see whether they can be used for diagnosis. Yes, now we have, based on the PCRs and all, is based on those regions only. And they have cloned that particular few regions which are missing in BCG and present in TB. So they have said this region is present in TB but not in BCG. Cut that and knock in into BCG. They found out that BCG started behaving like TB, and sometimes Stuart Cole's group, the same chap who sequenced this genome, we actually showed that there is a protection. So that is one vaccine candidate that is undergoing some kind of trials. And region 1 knockout of the knock-in of TB in BCG. Okay. So this is what I talked about. We'll just go through some of these. 5 bacilli gets put it into the macrophage, and then you have all these antigens and dead bacteria. They are taken up by immune cells called dendritic cells, go to the nearest lymph node, sometimes here, and activate T cells. These T cells are supposed to either help the macrophage get rid of the pathogen or kill the macrophage, depending on if it's a cytotoxic T cell or a T helper cell. Story looks extremely nice. Textbook immunology, whether it is TB or salmonella, this is what happens. Okay, irrespective of whether they are site, that's why I put site of infection. The problem comes here. TB ensures that every stage of this thing, that is what I have learned after 22 years of my research in TB. At every stage, it starts to outsmart our immune system. Something like this. When a pathogen comes, an antigen presenting cell or a phagocyte will catch it with a receptor, right? And take it in. TB modifies the receptor in a manner that even if it takes it in, there's nothing to do. then it is supposed to do a lot of stuff. Like for example, if a pathogen comes, time up.
No, sir, I'm just trying to start that.
Oh, it says
Presentation.
license बोल रहा है ना?
नहीं, मैं presentation start कर देती हूँ, sir. It will be easy.
Oh, I can use this pen on the
Yes,
presentation.
yes.
Oh, okay. That is it.
Slide. We will just put one.
Ah, yeah.
I can count. ठीक है. Okay, so just a second. I'll just put this way and then right click.
Thank you. Wonderful. Let's say here you have a bacteria. Then you have the macrophage or something, right? So this pathogen is picked up by a receptor. And then there is a formation of that. Phagosome, and then you have the bacteria. What is the next step? The phagosome has to fuse with the lysosome. Very good. Right? And the lysosome is supposed to kill it, and then you have the MHC molecules which will process and present the peptide so that the T cell responds. For this process to occur, the pH has to be less than 5.5. What does TB do? It secretes ammonia. Ammonium raises the pH, plus a few more proteins. This phagosome, there is a protein called RAP5, TTPase, RAP7. The phagosome is matured enough and to fuse this classical cell biology. RAP5 has to be downregulated, RAP7 has to be upregulated. TB prevents this step. So less acidification, absence of RAP7. RAP7 is like a marker for the lysosomes that now it's time for me to fuse with them. When there is no lysosome, there is no fusion. Okay, so that is how it stays put. Then some of our work identified various factors to show that it does what are the ways by which it does. So since we work a lot on calcium ions, for this to fuse, calcium ions have to be upregulated. TB ensures that calcium are thrown out from the macrophages. So it does all that. Okay. Then by using multiple mechanisms, by using one way force is calcium, all cells are born to die. If they forget to die, it becomes a tumor and then later a cancer. So the TB increases the lifespan of the macrophages till eternity. We showed that. And then using our model of HIV-TB co-infection, we found that these two join hands and increase the lifespan even further. Right? By how? By various mechanisms, down-regulating all anti-apoptotic proteins like BCL2, IAP, etc., and pushing up proteins like BAX and etc. Okay, IIF and all. So it does multiple mechanisms. How do I go back? Yeah. So that is there. Retention of RAP5, inhibition of complex molecules. Okay. Once a phagocyte means it has a phagocytic oxidase system. So a lot of ROS species, reactive oxygen species, reactive nitrogen species, it inhibits all that. Okay. So one of the molecules which can scavenge just like a sponge, ROS molecules is superoxide dismutase. Now this superoxide dismutase is made by the host. TB has its own SOD in its system. Not only that, using bioinformatics, we found out that TB, this is also known as molecular mimicry, right, in biology, where some of the molecules of the mammals, or let's say mammalian cells, are also present or mimicked by some of them. For example, cytokines. Many viruses have what are known as virokines. Right? Cytokine mimics of virokines. What will do? They'll simply go there and bind to the cytokine receptor and block its activity. Something similar occurs here also. So we found out that just like you have this like toll-like receptors, these are the phagocytic receptors, they have the signaling cascades. So once you have this, like we found out that TB has a protein, all these 4,000 genes that are secreted, there's one protein, my student Upasana, she worked on that. We identified a protein called RB3529C. Forget the name and all, it just comes automatically. This has a homology to one of the, for example, this is there. This is your toll-like receptor. So this has a signaling cascade. So one of the proteins, a scaffold proteins, known as MYD88, is a scaffold protein which acts as a docking site for many kinases so that the signal transaction can go down and diversify. It has a small domain to which one major protein called IRAC, interleukin receptor kinase, binds. This RB3529C, this is the TB protein, it has the same domain in its homology. So what happens? IRAC, instead of going there and binding and going through this thing, it simply goes and scavenges it here. So all toll-like receptor signaling gets inhibited. And some of the other things that, like the post-translation modifications like ubiquitination, stimulation, nidylation, changing of proteome, all these things are also going on in TB. Like we have shown with nidylation and stimulation also. It changes the cytokine balance. In COVID, you would have realized that there is a lot of inflammation, like IL-6, especially in severe TB COVID patients. So this inflammation is sometimes very good to take care of the infection. But in COVID, it was a major problem because it was so huge that the people were dying because of the inflammation rather than the virus. The viral load was lower, inflammation was high. So anti-inflammatory like hydroxychloroquine, dexamethasone are given. In India, using them is little tricky because we harbor this bacteria with us. So that is why they say, in fact, the government said that please don't self-medicate with all these things because this WhatsApp, a lot of stuff came, no? So they said that it has to be given very carefully to ensure that the right amount, it has to be subjective. The dose in one person may be different from the dose in the other person. It cannot be one tablet, 20 milligram, koi bhi kha lo. No. So otherwise, then you will get all sorts of other opportunistic infections will come because you are decreasing the inflammation, right? So your immune suppression may bahut zyada nahi hona chahiye. So all those things one has to take care of. So the cytokine balance is very critical. So what TB does is that it pushes down all inflammatory and Th1 protective cytokines and pushes up all anti-inflammatory cytokines. So I'll take few more minutes just to show what are the alternative strategies. This is all data, all published. Okay. One is that you modify BCG to improve it. For example, there is in addition to urea, to increase pH, it has urease enzyme also. So one group in Max Planck Institute by Stephen Kaufman, they developed a recombinant BCG. They removed urea. Delta urease means BCG, maybe urease hai, but both come compared to TB. So they removed BCG urease so that more phagosome-lysosome fusion. Now, and they put a listerio-lysine protein from Listeria monocytogenes. What it does, anything coming from outside generally gets processed on MHC class 2 and gives a CD4 response. You need a CD8 T cell cytotoxic response also. When you add LLO into this, it generally goes what is known as a cross presentation for immunology. And then it generates a CD8 T cell response also. So it generates both CD4 and CD8. Then BCG RD1, that means BCG in which the regions are different from TB. Your virulence region is cloned in so that to make it even more better. This is developed by Professor Anil Tyagi from South Campus, where he found out that if we overexpress this particular antigen 85C in BCG, it gave better responses than BCG. All these things are in the pipeline in various stages of clinical trials with limited success. So what is the strategy to treat them? Okay. What are the alternative therapies? One is drug repurposing. That means that's something that we in COVID we had hydroxychloroquine or remdesivir, things that were like tried out but nothing happened. Use a drug for some other ailment and see whether it works. Now the question is, how do I know which drug to use? Then what you do what is known as genome-wide RNA screen. Now everybody knows what is RNA interference? Yes? Everybody comfortable? Somebody people are just having half a nod or something. RNAi, everybody knows? Okay, fine. Say yes, no. So what you do is that these RNA libraries are available from commercially. So you have this, and when I say genome wide means there is an RNAi or siRNA, small sugar 21 nucleotide, against every gene in the human system. There are 30,000 genes are there. You take a macrophage, right, and then add 1. That way you do 30,000. So you have this 30,000. This is the last 5. So you have siRNA, SIG1, and then dot SI 30,000. Okay. So that means each time, so you do it in a 96-1 format or whatever. We have this well. Okay. So this is control siRNA, which means it's not against a genus scrambled sequence, nonsense sequence. That's a negative control. And this is siRNA 30,000K. So all these siRNAs are inside this. Let it go and knock down the gene of interest. Gene number 1 to gene number 30,000. Then you infect them with mycobacterial tuberculosis. Right? So now mycobacteria is going into a macrophage in which all the genes are there except 1. What do we need to do? We need to see whether that gene is important for mycobacterial survival or not. Then what you do is that after 72 hours or 48 hours, depending on the standardization, you lyse this and then you plate this for mycobacterial growth in your LB, 7-10 plates, and wait for 3 weeks, spray, and then wait for the no contamination. Then you look at the colonies. So you get a graph like this. So this is control. This is your scrambled RNA. TB is very happy. Some genes will be here, some genes will be here, some genes will have no effect. Like this. Okay, that means each of these line means that particular gene is absent. You look at this, which is a good drug target? The smallest one. Right? Now you know, and if it is going above that, that means it is very important for your TB growth. If it's not there, TB is even more happy. Balle balle. This is a problem. It is simply gone out. You identify the gene. When you look at the literature, is there any drug against this gene? Maybe for diabetes, maybe for cardiovascular diseases, maybe for neurology, right? Can we use that to treat TB? Nobody is going to say, then you do experiments. Nobody is going to say, you remove all these 20 drugs and just give this thing. You add on to that drug regimen. It is known as adjunct therapeutics, right? And this is what is known as drug repurposing. What is the advantage? Whenever you develop a new drug, identify a new drug molecule, like a chemist will synthesize a molecule, lot of things have to be done. Like phase 1 trial, phase 2 trial, somebody probably will talk about all that in your discourse. Right? You do that. It takes 20, 30 years, toxicity study. Remember, in COVID, they said that phase 1 trial, phase 2 trial, that is why they take 10 to 12 years. We didn't have that much time for COVID. That is why all these vaccines are emergency use. That means they said that this is an emergency, this is a global emergency, this is a national emergency. We have to save lives. So we'll see. We'll do a drug trial, vaccine trial, but may not be that extensive. If there are, let's say, thousands of patients have been given, they're working fine, go ahead, do mass immunization. Nothing will happen. So the mass immunization itself will give us a lot of information on that. And luckily, all the vaccines that are there, Covishield, Covaxin, they came out successful. So now we know COVID actually has taught a lot of things. We can teach from home, we can learn from home, we can work from home, and we need not wait for 20 years. Things have happened, and at the global scale, not in one Delhi or one like Karnataka or Tamil Nadu or Punjab or wherever. At the global scale, this thing. So now things are. Nucleic acid vaccines like DNA vaccines, RNA vaccines, people were scared of the ethics issue. Everybody, half the world has taken Pfizer's and Moderna's mRNA vaccine. Very happy. So that's what they're saying, that the next generation vaccines is going to be the mRNA vaccines. We just need to work about the stability because one requires -80, one requires -20. Struggling to keep a cold bottle of water in the fridge in a village, -80 to dur ki baat hai. -80 sterling here is also a major problem. So this is one way of drug repurposing. Based on that, we did a little bit of work and we found out that some of the antihypertensives using our work on calcium can be used as adjunct therapy. So some clinical trial is going on. I will stop here to take few questions or something. Yes, Shiva. You can ask some burning question in the lab also, but go ahead. I'm making.
Sir, we'll discuss that in lab.
Okay. But no, for the sake of it, at least to push up.
Okay, sir. Like, sir, I might sound childish, but sir, macrophage itself the reservoir for the MTP. Sir, they are containment zone for MTP. Sir, why our immune system again react the CD4 and CD8 and reactivate the macrophages? Instead, they could also activate the other phagocytic cells like natural killers and. So again, macrophages they can even sir like mycobacterium have sir mycolic acid that sir that also use like NH3 secretion could also increase the pH.
The table they go.
Again, macrophages.
Macrophage is there.
Yes.
You want the T cells to come here, the B cells to come here, and everything, right? Now forget the B cells, the killer cells and the NK cells.
Yes.
What does TB do? For any two cells to interact with, you require multiple receptor engagement. This is something that I teach in greater detail in the 4th semester advanced immunology called immune synapse. Just like you have neurological synapse, you have immune synapse. It's a very dynamic thing that processes. I mean, if I start, it will take another lecture, which Meenakshi will not allow me to. Okay, so all the receptors, there are about 8 to 10 receptors that need to engage with each other in the right ratios. So, for example, when you say NK cell, a natural killer cell, what is the evolutionary principle of a natural killer cell? Why do we have a natural killer cell when we have a CD8 positive T cells?
So they without any prior activation they can kill the tumor cells.
No. If tumor cells can be killed, we will be doing manga.
No, sir.
I'll tell you the problem. The first in evolution, I have to teach all these things next Monday. When this CD8 T cell comes in, what does it recognize on the macrophage? Which MHC? MHC class 1. Most viruses, HIV major culprit, downregulates the CD8 MHC class 1 on the macrophage. So CD8 T cell is completely blind to this. What did the evolution do? Evolution made another T cell called an NK cell, which looks for a cell where the MHC is absent. You must have read all nucleated cells have MHC. Why? All cells are not antigen presenting cells. It is to save themselves from the natural killer cells. Right? So that is one. But then that is like one statement for BSc level. In the MSc, we teach it both. It is not just the presence of the one MHC or there. Remember, in biology, I say that to my students, there is never all or none. It's there in physics. 2 plus 2 is good. The maths is good. 2 plus 2 in biology is Shraddha ki baat hai, kuch bhi le lo. It can be 22, it can be 0, it can be minus 22. With physics, if I throw this, it will fall down. In biology, it can go out anywhere. That is why standardizing something in biological systems is very good. Why do you think we don't have a for cancer? Cancer is some kind of an autoimmune disorder. We talked about tumor cells. No, cancer is some kind of an autoimmune disorder, right? Our own system is going haywire, cells forgetting. Pathogen is easy. It's an external environment, external stress. It's an external stress. This is an internal stress. My body is different from your body, from their bodies, right? So each of us have a different role. The problem with cancer is you're attacking your own cells, and you're looking at cells which have tweaked their genome a little bit. Any drug that goes here will kill the natural cells, normal cells also. That is why when a person gets diagnosed with cancer, there's not much problem. For example, in solid tumors, you see a lump here and there and all. Right? And with other circulating leukemia, that's different. The moment you start putting him or her on chemotherapy, the person is flat on the bed. Neutropenia, lymphopenia. Neutrophils are the first responders to any infections. they are the most sensitive to drugs, right? So they have to be very, very careful. So when you talk about macrophage, I'll tell you, interferon gamma is a major cytokine for activation of macrophages. How does a cytokine work? It binds to a cytokine receptor. Interferon gamma will bind to the interferon gamma receptor. It will not bind to the IL-1 receptor or IL-10 receptor. What does TB do? It downregulates the interferon gamma receptor on the macrophage. So even if you have a kilogram of interferon gamma on the cell surface, it is completely useless. TB patients make bucket loads of interferon gamma. Why is it not protected? Because there is no receptor. So there are 22 years I've learned how. So this is one of the major problems. Remaining questions we will discuss. Anybody else have any question? Survivor?
Not any questions. But very good, sir.
I don't have to say that. All yours, Meenakshi. Thank you for the message. I will. Where is the mouse? Yeah, you can just log out.
Yeah. Thank you, sir. It was really good. I really wish to attend your classes maybe for most of the you are alive.
Next week.
It's like long back I learned in biology and now it's coming up. It was brush up. It was really good.
That's why you use the term reorientation for us.
Actually, I told my student, I think one of them are logged in, but logged in के बाद कितना attend करना पता नहीं.
Ankush has logged
Ankush
in.
will log in, but ask him कहाँ था, sir मैं इधर था. If he is listening, he'll-
That is the benefit of online.
that is why the education suffered a lot.
I do agree. So now I call Astha from MIT University. She would like to give vote of thanks.
So we learn this also.
Oh,
Yes, Astha.
Thank you so much, sir, for the wonderful, enlightening, and enriching talk. We had a lot to learn about tuberculosis, and you made it very relatable for us using various examples and anecdotes, which for us it will um, lead to for development of a deeper appreciation for research and science.
that is
And I would like to thank our coordinators, Dr. Meenakshi and Dr. Rakarna, for very efficiently organizing the SERP programs and giving us the chance to present. And
No, they are brilliant.
thank you to our wonderful audience, respected teachers, PhD students, and my fellow SERP mates. I hope you had a wonderful learning experience today. So thank you, everyone.
Thank you very much. Right, Purna? This is mine, right?
Yes, yes. And coffee.
Okay, sure.